Okay, I think we should slowly get started. Good morning, everyone. Welcome to this morning session on connectivity modeling. We've got 6 speakers this morning, and the first talk is by Lawrence Frank, and he's going to talk about dynamic multiscale modes.
Thank you very much. So I'd like to talk about new method of looking at resting state data. So you're familiar with these resting state data, a typical set where every data point is characterized, if you look at one location there, by a spatial location and a temporal location. And you want to estimate the underlying signal, and it's in noise. And the real problem is that there's really no model for the signals, and the signals and the noise are nonlinear and non-Gaussian and overlap in space and time. So we're going to take a probabilistic approach, just the usual Bayes theorem, looking for want to estimate the signal given the data. And that's proportional to the likelihood of the data given a model and some prior information. Now we're going to assume that the signal, the underlying signal, is continuous in space and time. As you saw, each point was a space-time point. And so we're going to use space-time coordinates. And so you have these sampling points, but there's sort of an underlying continuous field, which is that orange surface there. And I refer to the sampling points as lattice points. Now there's a branch of physics called field theory, which is actually very good at handling fields, not surprisingly. And it's very good at looking at fluctuations and correlations and approximations. And it turns out you can formulate Bayes' theorem in terms of that. It's called information field theory. And H in that Bayes theorem is the Hamiltonian, which characterizes the conserved quantities. And the partition function actually generates all the correlations. So bear with me on one math slide. The Hamiltonian can be written in terms of the free, meaning non-interacting, terms, and the interaction Hamiltonian. And the free Hamiltonian is actually something you already know, and I'll show you that in a second. And the interaction Hamiltonian is what we will be interested in. We're not going to worry about normalization. There's an information source and a propagator. And just to show you that this is something you're already familiar with, imagine you had a very simple signal that had a known model function f, let's say it's the Fourier functions, and amplitudes a that you'd like to estimate, and you had Gaussian noise and no interactions. Then the information source is actually just a scaled inverse Fourier transform of the data. And the propagator is the correlation of the model functions. And the estimated amplitudes are just the propagated information source, which is, as you all recognize, the least squares solution. So you sort of know what the free Hamiltonian is, but that's not really that interesting to us. What we're really interested in is the interaction Hamiltonian, how things are coupled to one another. And it's a big mess. So it's over all space-time points. And actually, information field theory doesn't really tell you what to do with that. It's a huge parameter space. So a bunch of things coupled together, everything's connected to everything else, and it's not actually clear what the important part of that space is. So what we'd like to do is use prior information to reduce that parameter space. So the green dot is where I live in San Diego, and there's a ballpark. There's a little baseball down there. So there's a lot of ways I can walk from my house to the ballpark. But if there's city blocks that are blocked off in red, then that alters the pathways that I can take. And it turns out, actually, that knowing something ahead of time about what parts are accessible and what aren't tells you something about the pathways in any space or lattice. And that's called the entropy spectrum pathways, and that's something we developed. So let's take a really simple example. Imagine those white squares are rooms that you can go into. The black squares are rooms you can't get into. And what we want to do is we want to have a coupling matrix that describes motion in that space. So if I can move from I to j, they're both white, then qij is 1 in this case. And I to k, I can't get in the k room, so that's 0. And so you recognize q as the adjacency matrix. And the interesting thing is that the eigenvectors of q tell us about the most accessible regions of the parameter space. And why is that? Why is that? Because everything related to the next thing tells you about something about the pathway in that space. And this is actually an incredibly powerful concept because if you just make a bigger version of that lattice, accessible, non-accessible, and you calculate the adjacency matrix of that, then the principal eigenvector that tells you where the equilibrium probabilities are, and that's the bright spot in your eye. I don't have a pointer that works, so you can see that there's open spaces that have higher probabilities because there are more pathways there. This is actually called the maximum entry random walk discovered by Berta, but it actually turns out it has nothing to do with a random walk. It simply has to do with prior information. So the ESP theory really is that Q is any coupling matrix. And the important point is that coupling matrix is user-defined. So whatever problem you have, if you can generate Q, then you can use that. And in fact, we use this for our diffusion tractography, where we use local coupling information to generate global tracks. So that lattice, it's a very general concept, this ESP concept. So it can be any coordinates. So space-time coordinates are what we're interested in from the very first slide. So the entropy field decomposition is information theory, but it uses the entropy spectrum pathways to convert space-time coupling information to figure out what the most probable configurations are. And there's no assumption of Gaussian signals or noise, linearity, etc. So here's a schematic. We had that ridiculous coupling matrix. It was huge. Using coupling information, we can reduce that space and have the Hamiltonian depend just on the eigenvectors of the coupling. And from that allows us to solve for the field modes, which is what we're after. So here's an example. This is a very simple example. Two overlapping ellipsoids that have different time periods, and the overlapping part has a very complicated signal, or more complicated signal. Independent components analysis fails on this, and this is well known, but it's a good example because it's actually fairly simple. The two modes detected by EFD are two space-time modes that easily separate. Here's a more complicated example where we have overlapping non-periodic ellipsoids, and the different colors correspond to the different detected modes. And so the fact they're all different colors means that the EFD has detected those. And here you see those detected signals. So here's some data, NYU test-retest data. On the left is trial 1, on the right is trial 2, and this is pretty typical of EFD results on a single subject. You get very well-localized activation regions. Those are not processed any more than what I just described. There's no filtering of the noise or anything funky like that. So it shows you that you get very clear activations on a single subject. By the way, these were registered using a new registration algorithm that's registered different modalities, and that's poster 4054. Here's some single subject results from data from Tom Liu at UCSD and ICA compared with EFD and default mode. The names of these modes, by the way, are the ones that are closest to the ones that you see in the literature, but I think the most interesting thing is that you see lots of spots show up in individuals which are not perhaps exactly like the salience network, for example. And it seems like those are probably, in some ways, the most interesting because they vary amongst individuals. If you average over subjects, you get things that look more like what you see in the literature because you've averaged over all the interesting little spots. And here's some other well-known modes. So here's an example, just one mode, and you can see that it's a non-periodic, very complicated activation. Now there's a bonus here, which is kind of interesting, which is that in order, we're looking at coupling information, space-time coupling, so part of the calculation that we have to do is to calculate space-time correlations. And so we're left with a space-time correlation tensor. And so what you can do is you can use these to define another Hamiltonian. You can just put these into your tractography algorithm because our tractography algorithm is using tensors. And so we have something we call functional tractography. So what that is mapping the maximum space-time correlation. This is a single mode of a single subject. So this seems like quite an interesting way to look at connectivity. Here's another example. And as usual, what do you do with tracks? One of the things you can do with tracks is you can calculate the connectivity matrix from every voxel to every other voxel. And if you calculate the eigenmodes of the connectivity matrix, you can generate what we call functional eigentracks. So this is the principal eigenmode from the functional tractography. And I think this is probably, or hopefully, another useful way to look at data. So I would just want to finish up by saying that all this is freely available. We have a platform called Quest, which is a beta version, and it actually does the diffusion stuff that I showed and the functional stuff, and does all the warping and everything else. So feel free to visit that website, and if you'd like, download it. And we'd like some feedback. For the final slide, I just want to put some real numbers on here. This is how long it takes to do all of this stuff. This is everything that's done in Quest, but I've circled the ones that were shown here. So that mode estimation took about a minute, and the tractography took about 3 minutes per mode. Registration's about a minute. So it's all, and those were actually timings from my Mac laptop yesterday. So it's very, very easy for people to use. And in fact, if you have a Mac, you can just drag and drop the application. That's it. Thank you very much.
Thank you very much. We've got time for one question, if there are any questions. I was interested to look at the sort of spatial layout of the modes that you were showing. Mm-hmm. So it seems like they have a very focal center and then a sort of quite ellipsoid or round shape. Is there any reason for that in your modeling, or do you think?
You mean just the normal modes that we see? Yeah, those were, when you actually register those, they actually are more well confined to shapes. Right now, that's mostly resolution related. Right. But when you do it, you actually see with that nonlinear registration routine, so typically when we do it, when you register it, you actually see it very well localized within gray matter regions. Within gray matter.
So, okay, that's really nice. Okay, thank you very much. If there's no more questions, we'll move on to the next speaker. So the next speaker is Chao-Gan, who is going to speak about concordance among indices of intrinsic brain function.
So good morning, everyone. It's my great honor and pleasure to introduce our work here, concordance among indices of intrinsic brain function. So when we're doing nothing, we are not doing a specific task. The brain has lots of intrinsic brain activity. So you can see here, some regions the activity goes up, goes down, and interestingly, you can see some regions that goes up together and it goes down together. They form some networks. So tons of methods have developed to characterize this intrinsic brain activity, like a degree centrality, like a regional homogeneity, like a fractional amplitude of low frequency fluctuations. There's a very long list of these kind of methods. So given time limit, I cannot introduce them all. But there are some I would like to introduce. For example, to measure a voxel strength, so we have a voxel, we have a time series, you can convert it into frequency domain and measure the power or amplitude. We got amplitude of low frequency fluctuations, or we call it AFF, or the fractional one, FAFF. And also, we have a voxel. We can measure its concordance with its neighbors. So the local synchronization, or what we call it the regional homogeneity or RHO. And also, if we have a voxel, we can have its counterpart in another hemisphere. We can measure the homotopic connectivity, or we call it voxel-mirrored homotopic connectivity. We can also measure global connectivity. We have voxel. We characterize its connectivity with every voxels in the brain and count the significant connections. So we call it degree centrality. Another way is calculate global connectivity is we get a global signal. we calculate the correlation with the global signal and the average voxels. We can use this method to define how a voxel follows the brain trend. So we have lots of methods. These kind of methods, they are definitionally distinct. But do they capture unique variance, or do they have lots of shared variance? What's their interdependencies? These questions still remain unknown. So we can measure the concordance of the methods maybe across voxels, what's their interrelationships, or across individuals, or even across time. So there are some initial studies. So they measured the correlation between regional homogeneity and the amplitude of low frequency fluctuation across voxels. They are highly correlated. There are some other studies measured across individuals. So regional homogeneity and degree centrality. So in an individual, in a voxel, if one subject has a high ReHo, we also have high degree centrality, so as ReHo and FF and the degree centrality. So this kind of studies have measured some of the interrelationship, but not full. And also, what's a relationship across time is not unknown. So the goal of the present work is give a comprehensive examination of the concordance among different intrinsic brain activity measures, both between and within individuals. So we would like to ask, what's the concordance at the global level? So both across individuals or across time. And the second question is, what's concordance at the voxel level? For each voxel, what's the concordance for that voxel? And what's the concordance at the volume level, so across space? And the last question we would like to ask is that concordance have any neurophysiological meaningful? So we examine the age effect on the concordance. So to answer these questions, we use an enhanced Nathan-Klein Institute rock-length sample. That sample has a multi-band sequence, so the TR is 645 milliseconds, so it's pretty short TR, so it's good for temporal dynamic analysis. We use 173 neurotypical individuals aged between 8 and 6. That's a lifespan sample. So we examined 5 alpha indices for voxel strength is FFF, regional synchronization is ReHo, homotopical connectivity in the VMHC, and the global connectivity is degree centrality and the global signal correlation. So for the first question is, what's the concordance at the global level? So we measure the correlation between global mean of the alpha indices across individuals. For a given individual, have a AFF map, we calculate the global mean. So across individuals, that's a series of the global mean of FFF. And the ReHo, we have a series of global mean of ReHo across individuals. So we can calculate their cross correlation. So we have 5 measures. We can have a 5x5 correlation matrix. Here is the correlation matrix looks like. So actually, all the 5 measures at global level, they're highly correlated. So what about across time within individual? So for a given participant, we have used the sliding windows. For each sliding window, we can calculate the FFF, or regional homogeneity, and the other methods. And for each individual, have a series of FFF maps. So we calculate the global mean. We have a global mean time series, and so as a ReHo, and we calculate the correlation. So for each individual, we have a correlation matrix. So here on the right is a within subject. It's across time correlation. It highly resembles the between subject results. And we also ask if this high concordance is contributed by the nuisance signals, like head motion, like global signal, like respiration, like heart rate. And actually, you can see here is all the nuisance signals is not contributed to this high concordance. So it seems underlying this high concordance, there may be some underlying neural signals to driving all the functional aspects. And the second question is, what's the concordance at the voxel level? So we calculated the voxel-wise concordance index across individuals. So for participant, we have 1 voxel. So the 1 voxel has a value. So across individuals, that voxel, we have a series. And for ReHo, we also have a series for that voxel. We can calculate that. So we have 5 series of that. We can calculate the concordance using Concordance W. So we can define concordance map across subjects. So basically, this concordance map, we see it is have a very high concordance in the gray matter voxels. So all the cortex have very high concordance, but the white matter and CSF have very low concordance. We also look at within subject across time. So for subject, for each time window, for each voxel, we have a value. So across time, there's a time series for FFF for that voxel, and also ReHo, also degree centrality, VMC, and global signal correlation. So we have 5 time series. We use Concordance W to measure their concordance. So we can have a concordance map for each individual. So we perform t-test on that maps, and we have a group map on the right. The concordance map, the temporal concordance map, looks very similar as a group, as across individual results. So in the white matter and CSF, they have very low concordance. So probably these regions, because they do not support function, they have more noise at the different function aspects, so they have very low concordance. And also, for both between individual results and within individual results, we use a rank order. We found that very similar. They are highly correlated. So the between subject results and the within subject results is very similar. The third question is, what's the concordance at the volume level that's across space? So for each individual, we characterize the static volume-wise concordance index. So for each individual, we have an FF map, we have a ReHo map, we can calculate the spatial correlation, or we have 5 maps, we can calculate their spatial concordance. So for each individual, we have 1 index, that is the concordance index. Across subjects, we have a series. We also measure the dynamic volume-wise concordance index that's across space as well. So for each individual, for a given time window, we measure the spatial concordance of the FF maps, ReHo maps, degree centrality maps, and we can have, for each window, we have an index. So for a given participant, the concordance goes up sometime, goes down sometime. And for insanity check, we just measure the correlation between the static concordance and the mean dynamic concordance. They are highly correlated. So we would like to ask for the difference between the high concordance windows and low concordance windows, or between the high concordance subjects and low concordance subjects. What's the difference? So we performed 2 sample t-tests, just to divide them into high concordance subjects and low concordance subjects. We found for every aspect, for FFF, for the regional homogeneity, for degree centrality, all the different function aspects always have higher values in the high concordance subjects. This also remains true within subject analysis. Within subject, we have high concordance windows and low concordance windows. And in the high concordance windows, like regional active strengths like FFF, and global synchronization like global degree centrality and global signal correlation, they are all high. So it seems in the high concordance states, the brain is highly connected, so it's a highly integrated state. So the first question is age effect on concordance. We measured on the voxel-wise concordance index. Almost all the brains showed a negative correlation with age, and when we average across the brain, the concordance decreased with age across the lifespan. So this is for voxel-wise concordance index. We also measured the volume-wise concordance index. It also drops with age. This is for static one, and we also calculated for the mean dynamic one. So all the concordance decrease with age across the lifespan. Some people suggested as aging, the brain is less connected. So this disconnection maybe has some function of this concordance decrease. Or some studies suggested across the lifespan, no matter it's during development or during aging, the cortical thickness always decreases as age increases. So probably the cortical thickness decrease, there are less functional units, so that will decrease the concordance. So basically we just provide a comprehensive examination of the interrelationships of the alpha indices. They showed a high degree of covariation, and high concordance always related to the high strength of all the various alpha indices. And the concordance of the alpha indices negatively related to age. So I would like to thank my collaborators and the funding. Thank you very much for your attention.
Thank you very much. We've got time for 1 or 2 questions, if anyone has any questions. Maybe you could explain a little bit. So I was quite surprised to see the high concordance because some of these measures are indices of quite different things, like regional homogeneity being a regional measure, and other measures like degree centrality being much more of a global measure. So were you surprised to see that, and what is your hypothesis of why?
So we speculate that we expect the high concordance may be related to some inter-network is increasing and the intra-network is decreasing. But we also performed that kind of analysis, both within network and between network are increased. So we suggest in the paper this may be some high energy states, but we do not have electrophysiology signals to characterize that.
Okay. I thought your age correlation was very interesting, but did you look at whether there was some part of that was explained by increased motion or something like that?
We've looked at motion that cannot be explained by motion.
Okay, so that's completely independent from potential increases in motion. Okay, that's great. That's really interesting. Okay, thank you very much. If there's no more questions, we'll move on to the next speaker.
Okay, thank you.
Thank you. So the next speaker is Nicolas Farrugia, and he's going to talk about identifying spatiotemporal patterns of functional connectivity with dictionary learning.
Thank you very much. So I'm really happy to be here to present this work today. So we're trying to identify spatiotemporal patterns of functional connectivity, and especially we're interested in dynamic aspects of connectivity. So most of you will probably be aware of the sliding window k-means method for dynamic functional connectivity, but I just try to recap quickly what it consists in. So you get a set of time series from resting state, typically, and you use a set of sliding windows to identify temporal covariance matrices unfolded in time. So you have a series of temporally consecutive covariance matrices. And the k-means method is just doing a k-means clustering to try to identify recurring patterns. So this corresponds to centroids of covariance matrices. And people have interpreted this as dynamic connectivity states that you can then trace for single subjects. You can trace which states people go through. So this is a very promising method that's been used in many aspects in the literature, also in task data. And we're trying to go slightly further of this method, trying to extending the sliding window methods to address two main limitations. So the first thing is that when you use k-means clustering, you basically project the current state into only one state, because k-means is just a method which is a clustering method and not a decomposition method. So the current state is the closest state in terms of distance to the centroid. And the other limitation, the second one, is that the observations that are fed into the k-means are considered as temporally independent, which means that you have all this set of the consecutive covariance matrices, and you could completely shuffle them in a different order. You would get exactly the same result as the output of the k-means. So the states that are estimated don't depend on the temporal dependencies of one covariance matrix to the next. So there's two different ways that people have tried to address these limitations already. So as an alternative to the k-means, the group from Cairo has tried to use temporal ICA to identify overlapping states. And the output you get from this method is a weighted average of covariance matrices, so all the states, and they've been called meta or multi-states. So this is an interesting method, but it relies on the assumptions of ICA, which is temporal independence assumptions. And the other way to address the second limitation, so the fact that the covariance matrices are temporally depending on each other, is to use, for example, quasi-periodic patterns. So this is the work from the group from Kyle Oss that's been also presented previously in OHBM, which consists in taking a chunk of consecutive temporal matrices and identifying recurring patterns in the whole sequence. So we're trying to combine the best of both worlds of these two approaches by using dictionary learning. So the proposed method will use sparse online dictionary learning, which is a metric decomposition method with few assumptions, and is basically trying to learn a base on which you can decompose the data with sparsity constraints. And we're using this method on sequences of timing windows, of covariance windows. So I'm going to describe this method graphically in the two following slides. So The input is the same. We use sliding windows on the time series on the brain parcellation. So we estimate connectivity using the multiplication of temporal derivatives, which is a measure that's been proposed last year by Jens McShine, which is basically a better measure if you want to use short sliding windows, and it's more sensitive to transitions and to both transient and stationary transitions in connectivity patterns. So we use this measure, and we get a set of MTD matrices in time, and we feed this into this dictionary learning, but we use sequences of consecutive matrices, so which means that we take into account the dependency, the temporal dependency between connectivity patterns. So the idea is that the output you get is of size k by 70 by 70, with k being the length of the temporal pattern you're trying to extract, and 70 is the number of time series in this case. So the dictionary is a set of spatiotemporal atoms that are representing typical transitions in connectivity. And we now need to know about the states when they're active in the signal. So we try to estimate their time series. And to do so, we use convolution. So we basically use a full convolution of each atom on the original set of covariance matrices, and this gives you peaks of activity where each atom is active. And in order to summarize this signal further, we use a second dictionary learning, which extracts recurring pattern of this activation state. So this is useful to further summarize the connectivity into what we call in the end dynamic functional connectivity states, which is a set of spatiotemporal maps of size k by 70 by 70. So you can see that as a set of 3D covariance matrices, so transition in connectivity patterns, and each of these states has a time series, which we express in terms of a relative ratio, which is a number between 0 and 1. When it's 1, it means that only this state is active currently, and 0, which is not active. So it's summed up to 1. So I'm going to present some data that we have run on a sample of a healthy subject with 4 resting state scans. So it's a test retest with 84 participants. And the data was parcellated, projected on the cortical surface, and then parcellated with an anatomical parcellation. And we applied our method with the following patterns. So we used this window of 15, which is the size of the temporal pattern we're trying to extract. We used 6 atoms and 4 states. So these 2 parameters have been chosen using a stop criterion. So we're just adding atoms, and then we stop when there is no more significant gain on explained variance. And in the final result of the states, so you still have to cluster the rows, so the order of the rows in order to identify networks. So this was done using k-means clustering. So we're grouping the resulting rows into networks that are coherent in terms of spatiotemporal dynamics. So this is how it looks like. So here I'm showing you states 1 and 2. So you have to remember that each state is a sequence of connectivity matrices, so MTD matrices in this case. So on the left column, you can see the temporal average over the 15 matrices, so over the sequence. And in the middle, you have the STD. So what you can see with this thing is that you get networks that we have named with the reordering of the rows, and you can see that in the average you have some within and between connectivity of the networks, which don't necessarily correspond to what you get in the STD, which means that we have identified patterns of transitions between 2 networks, or more than 2 networks in certain cases. So these are the 2 first states, and on the right side you can see the time series of the relative ratio for the states, which corresponds to how much active they are. So you can see that state 1 is around 0.8, which is it's a prominent state, and state 2 is around 20%. And I'll quickly show you the 2 other states, which are 3 and 4. So they are more sparse state in the states are more sparse in their, especially state 3 in its connectivity map. And you can see that they are less active, which means that they are more correspond to fast transitions in the connectivity pattern. We've tried to characterize the activity ratios of the 4 states, so using simple kernel density estimation, which is Gaussian mixtures. And you can see that state 1 is clearly prominent with an activity which is centered around 80%, of like a ratio of 0.8. And state 2 is around 20%. So states 1 and 2 are probably the most important ones, and the other ones are less active, but are still correspond to peaks of activity in the data. We've also looked at consistency, so the test-retest, across 4 scans. So what you see here is consistency of the activity ratio. So this shows that across the 4 scans, it's always, the state 1 is always the prominent one, and the states 2, 3, and 4 are also quite reproducible in terms of how much the activity are spread. And we also looked at consistency of the spatial maps. So we use the cosine similarity measure, which is maximum. When it's 1, it means it's perfectly similar. And you can also see here that you have across scans state 1. You always get a good correspondence between states 1 and 2. You can always find a good correspondence between the states. You also have some intra-correlation. So as a conclusion, we extracted dynamic functional connectivity of states that can overlap. So we identify overlapping states which are active at the same time, and we estimate the time series. So the other interesting thing in this work is that we estimate transition between connectivity patterns. We have a temporal dimension, and we have evidence that these states are reproducible as well. In next step, we're going to validate this using artificial data and also trying to vary the size of the sliding window, so the temporal dimension. And we have also the same data. We're looking also at EEG and task data to try to make this work, to identify transitions between states between active states. So I want to acknowledge the funding, which is coming from the Brittany region in France, and this work would not have been possible without the collaboration with the Max Planck Institute and the group of Daniel Margulis in Leipzig. And this work is also presented as a poster this afternoon, so I'll be upstairs. And we have an opening in the lab to continue working on this work. So I thank you for your attention.
Thank you very much. We've got time for a couple of questions, if there are any questions. Yes?
A number of comments. What you're trying to do looks really interesting, particularly the dictionary learning and your attempts to get at the variability of the results. That's great. It would be good if other people working in this area took some hints from that. One technical comment. So the people that do dictionary learning seem to be in love with this algorithm of MyRAL, but there's a problem. It's an online algorithm. So at any point in time, it's only using information available up to that time, whereas you're doing an offline analysis. So the sliding window technique, for all its miserable misuses, is actually using information from 2 sides of the time point. So the question is to develop a dictionary learning procedure which would be using information from both sides. So if you like, anti-causal. Actually shouldn't be too hard to do something, but would be worth looking at, I think, because the obvious point is with a limited amount of data, you would like to use as much information as you can.
Very good comment. Thank you for this. And it's a good idea, I think, to try to rework on dictionary learning itself. I mean, we're still using, the second sliding window is still using a sequence of the data. But we actually use, so that was to reply on the causality comment, but you would suggest to use it in both directions. But actually, I didn't present the detail here, but we actually use the online version. And we re-estimate the error globally per subject and for all subjects at the end by re-encoding the data and estimating the error. So we are taking into account also multiple subjects and different windows in the subject. But I agree with you that it would be interesting to try to develop a specific approach.
Thank you very much. We'll move on to the next speaker then. Thank you. So the next speaker is Valerio Zerbi, who is talking about the structural basis of large-scale functional connectivity in a mouse.
Thank you. So in the past year, there have been a lot of numerous studies on connectivity and connectomics, And this is due to the fact that many technologies have become available to map the connectome either functionally or structurally at different scale level. However, when we look at studies on human connectomics, the vast majority, if not all of them, are done at the microscale level using MR imaging. And MR is also basically the only available option that we have to measure both the structural and anatomical connectome using diffusion-weighted imaging and photography, and also the functional connectome using resting-state fMRI. So these methods are very interesting and powerful. However, when we want to compare them with something more biologically relevant, we found out that there are some certain fundamental limitations of these approaches. In particular, like both of these methods rely on indirect measures, either of water diffusion or bold activation. And even more, like diffusion-weighted imaging and photography suffer from a very low sensitivity in the gray matter, which is the region we are actually mainly interested in. And despite what you might have heard at this meeting, even the best photography algorithm will have problem in resolving crossing fiber and will not tell you where exactly a fiber starts and where it ends. So while the diffusion-weighted imaging photography can be still considered as a work-in-progress tool, the resting-state fMRI, it seems very robust in measuring connectivity and with like even between subjects, within subjects, and even across species. Still, when we want to compare the function with the background anatomical structure, we need, therefore, to compare the resting state data with something which is more biologically relevant, like more close to neural connection. So in this study, we scale down our functional to structural connectivity problems with, on the simplest animal model, in which we could have both the resting state of MRI data and an anatomical ground-truth of structural connectivity. And we found this model in the mouse. Because actually for the mouse, we do have a mesoscale structural connectome available, and this is from a work from the researcher of the Allen Brain Institute, published already 2 years ago, and this is based on more than 2,000 viral tracer injection. So these are monosynaptic anterograde anatomical connectivity. And in our lab, we are specialized in acquiring and processing resting state fMRI data in the mouse. As you can see here, so we previously published a work where we show resting state network identified in both the cortical and also subcortical level with very high similarity to the one that are normally found in humans. So in this work, we wanted, therefore, to compare our data with the background, with the structural connectome provided by the Allen Brain Institute. So we acquire resting state data on 14 black 6 mice using a 9.4 scanner equipped with a cryogenic dedicated mouse brain coil. We use a mechanical ventilation for the best delivery and control of the anesthesia. And we use a low dose of isoflurane combined with methamphetamine. And for the acquisition protocol, we use a multi-echo gradient echo sequence, which allows the semi-automatic regression of the non-neural component from your signal. This is our method overview. So basically, we downloaded from the Allen mouse brain connectivity dataset all the injection done in black 6 mice in the isocortex, hippocampus, cortex, subplate, striatum, pallidum, and thalamus, which are the regions that are fully covered in our field of view in our resting state fMRI dataset. We then normalized everything in one common anatomical template based on the from the Australian Mouse Brain Institute. And then we took the same seat coordinates and the same seat size of the tracer injection to create 238 seat-based resting state fMRI experiments. And this is how an individual result looked like for one injection here in the somatosensory barofield cortex. You see in green the tracer intensity projection, and in red the resting state fMRI connectivity. You can already see that there are very much similarities between the two metrics, particularly in the contralateral cortex or in the striatum, but like in the thalamus, like here with the red arrow, for example, we could see some areas in which we saw a structural connection but not a functional connection. So when we then compile all this information together, all these 238 experiments together, we then could build up the macroscale functional connectome for the mouse brain. And we could finally then start identifying regions of similarity and difference between known long-range connection, for example, the connection from the cortical region to the contralateral cortical region, and we found quite a good agreement between the function and the structure. And this agreement was also found between the cortex and the ipsilateral striatum connections. But for instance, when we look at cortex to striatal connections, the high and strong connectivity of this, is identified from the structural connectome was not mirrored by a functional activation. We then want to see even more in detail if the structural projection from the cortex to the rest of the brain were actually mirrored by a functional activation. We did that by using a winner-takes-all approach. So we selected 20 different seeds in the cortex, we color-coded them, and then we basically measured the strength of the connectivity in every other voxel in the brain. And by doing that, we could then compute what are called the tomographical maps. And these are based on the tracer injection data, so all the data is already available, but since we have everything in the very same space, we could also compute the same tomographical map for the rest state fMRI data, obtaining a quite striking similar maps. And we also performed a statistical analysis on each of the voxel, finding out that almost 87% of the contralateral isocortical voxels showed a significant Spearman correlation, and this is corrected by a false detection rate. And of course, we did it with all the other structure, and here, for example, we could see that almost 88% of the striatal voxels showed significant Spearman correlation, while only the 8%, almost 8 or 9% of thalamic voxels showed a significant correlation. Finally, with the help of a graph theory approach, we measure the minimum distance between each of the nodes in our matrices. And we, therefore, by doing it with different thresholds, we measure what are called the sparsity profiles for the functional and the structural matrices that you can see here. And you can see that already there is quite some good agreement between the sparsity profile. But what is most interesting is that now we can interrogate these matrices. For example, we can see which are the nodes that are connected by only one edge in both the functional and the structural matrices. And with a particular interest in interhemispheric homotopic region, we could see that, for example, regions that are connected by only one edge in both the functional and structure are basically cortical homotopical region, while instead regions that still have one edge in our functional matrix, but more than one in the structural matrix, are mainly derived from subcortical pathways. And we call them so we can basically identify polysynaptic and monosynaptic functional connectivity pathways. So to conclude, we report here brain-wide microscopic functional connectomic for the mouse. All the raw data and the seed-based maps will be freely available, and I believe this is an opportunity to test new paradigm or new models or new approaches to investigate resting state fMRI. And I see the people that spoke before me, so that will be a very good data set to work on. We demonstrated the cortico-cortical and cortico-striatal circuit. They have a very high correspondence between the function and the underlying monosynaptic structural wiring pattern. This basically gives a window of confidence of which are the circuits that we can target with our resting state fMRI approach. And I'm thinking about models for autism, Parkinson, or even schizophrenia, as we've seen this morning. And by contrast, certain subcortical network, like the left and the right striatum, do not share a direct anatomical connection. Rather, the cultivation, we believe, is more coming from a top-down cortical control. And finally, the lack of cortico-thalamic connectivity still remains a matter of investigation. We believe it might be an effect of anesthesia, but we are exploring also other hypotheses. So with this, I would like to thank the people that have been working on, and if you want to discuss it more, I will be this afternoon at posted 3941. And thank you.
Thank you very much. It's time for a question. Anyone has a question? I was curious about the monosynaptic versus polysynaptic connections and relating that. I was wondering if you'd looked at, so you're using full connectivity for the functional connectivity analysis?
Yes.
Did you look at partial correlation at all?
I did look at partial correlation, but I realized that while using partial correlation, especially when you have hubs region, regions that are connected to multiple areas, you try to basically emphasize the connection that are the strongest one, but you then miss some other type of connection within the same region. So therefore, we decided to go just for functional, for the total correlation.
So your results are showing that there's better correspondence for the full connectivity rather than the partial connectivity. Yeah, that's really interesting. Okay. Thank you very much. If there are no more questions, we'll move on to the next speaker. Thank you.
Thank you.
So the next speaker is Linda Geerligs, who is talking about multivariate distance correlation being a more robust and reliable measure of functional connectivity.
This is not my talk.
Can we change the slides, please, to the right talk? Sorry, these are not the right slides. If someone could change them around. There we go.
Yeah, great. Okay, so today I'd like to introduce this new measure of functional connectivity called distance correlation. So many of analyses of functional connectivity, they are based on regions of interest. So we have these regions of interest scattered across the brain, and we look at the connectivity between each pair. Typically what we do is that we average across all of the voxels within a region of interest, and then we compute the Pearson correlation between any two pairs of regions. But this method is probably not the most optimal way to do this for a number of reasons. So the first reason is that we know from techniques like multivoxel pattern analysis that there is a lot of information contained in the individual voxels within each region of interest. So here I'm just plotting voxel by voxel correlation matrices for two ROIs, and these are very small ROIs of approximately 20 voxels. So you see that in some of these examples from real data that the ROIs are quite homogeneous. But in other cases, like the one that I drew the square around, you see that some of the voxels in ROI2 correlate positively with the voxels in ROI1, and some correlate negatively. So if we use our typical approach of averaging across voxels in each ROI, we wouldn't be able to detect any connectivity here. The second potential disadvantage of the current methods is that we're only able to detect linear associations between different brain regions, and we might miss nonlinear interactions between regions. So the measure that I'd like to introduce today is called distance correlation. And distance correlation is different from Pearson correlation in three important ways. So first of all, it's able to use information from all of the voxels within an ROI. Second, it's able to measure both linear and nonlinear associations. And third, it cannot differentiate between positive and negative correlations, and that's because of its multivariate nature. So just to try to briefly illustrate how we can compute distance correlation, how this works. So here I'm starting off with a simple sine wave. And the first step in computing distance correlation is converting this sine wave to what we call a distance matrix. So what that means is that for each pair of time points, we're computing the distance between them. And in this way, we build up this matrix of time by time. So if we have two time points in our sine wave with very similar intensities, we have a low distance. And if we have two time points with very dissimilar intensities, we measure a high distance. So from this is just an example of a univariate case, but we can also expand this to a multivariate case. So here I'm just showing you two ROIs, and each ROI contains two different voxels. And now, for each pair of time points, we can now compute the total Euclidean distance between all of the voxels at time point 1 and all of the voxels at time point 5, for example. And we go on doing this until we have all of the pairs of time points in our distance matrix. And importantly, we do this separately for the two regions of interest that we have. So we get these two time-by-time distance matrices. Then in the next step, we center these matrices. And then the important step is that we can then look at the variance and the covariance of these distance matrices. So you can imagine that if two distance matrices have a high covariance, that this means that the distances change over time in a similar way. So this can then lead us to conclude that they are functionally connected. So in actually calculating the distance correlation is then very similar to a normal correlation where we just divide the distance covariance by the distance variance. So I've done some simple simulations just to illustrate some potential advantages of this method, or at least differences with the traditional Pearson correlation. So here is our baseline case where we have this perfectly homogeneous ROIs. So all of the voxels within ROI are strongly connected, and we have strong connectivity between the ROIs. So here on the left you see the Pearson correlation and the right distance correlation. Now, we compare this to a case where half of the voxels are negatively connected to the voxels in another ROI and half positive. So in this very extreme case, we find no correlation at all with Pearson correlation, and we find the same as our baseline example with distance correlation. In a somewhat less extreme example, we see that half of the voxels here are connected to the voxels in another ROI and half are not. And here we see that, again, that distance correlation is more robust against this inhomogeneity. So the difference between our baseline case and case 3 here is smaller for distance correlation than Pearson correlation. So then we evaluated this measure of distance correlation on real data. So we had repeated measures resting state data from the Cambridge Center for Aging and Neuroscience. So we had 236 participants. And we looked at three different sets of regions of interest. So we had AEL ROIs, we had the power ROIs, and we had a set of Craddock ROIs. So let's first look at the connectivity matrices for these different, for the Craddock ROIs. So here is the connectivity matrix just with our typical Pearson correlation approach. So these regions in this matrix are ordered by the network. So on the diagonal, mostly within network connections. And the color indicates the strength of connectivity. Now I can compare this to our distance correlation matrix, and you see that it looks very similar. So we find a very similar network structure overall. Now we can also look at the difference between these two, and then we find that it's actually mainly the subcortical regions here showing big differences between these two measures. So I've divided this connectivity plot here into these three sections with the black lines. The top section are sensory motor regions, the middle section are subcortical regions, and then the bottom section are regions involved in higher order networks. So you see that it's mainly these subcortical regions showing a difference, and this makes sense because these subcortical regions are also the ones that tend to be least homogeneous. So these are smaller regions with more functional specialization on the small surface. So basically, if we plot an ROI on top of that, then we are measuring a lot of different functional systems within one ROI. So our next question was then, is this difference because our measure is multivariate, or is it because it's nonlinear? So to answer this question, we then looked at a univariate version of distance correlation. So we first averaged across all of our voxels within an ROI, and then computed distance correlation again. So here, if we look at the difference between this univariate version of distance correlation and Pearson correlation, we see that there are no marked differences between the two. So we can conclude that it's actually the multivariate nature of the method and not the nonlinear nature that's causing these differences. So next thing, we looked at the reliability of our connectivity estimates. So to look at this, we looked at the correlation between each participant's connectivity matrix at time 1 and their connectivity matrix at time 2. So each dot in this graph is one participant, and the blue is the distance correlation results, and the red the Pearson correlation. So we see that for each of the 3 sets of ROIs that we started with, we find a significantly more reliable connectivity matrices with distance correlation. And again, we can ask, why is this? So one potential option would be that because we're not taking the sign into account with distance correlation, that this is one of the reasons for change in reliability. So we can look at the unsigned version of Pearson correlation. And if we do that, we see that this actually can't explain the benefits that we see with distance correlation. Next, we can also again look at this univariate version of distance correlation that I talked about before, and you see that only in the case where we have this multivariate distance correlation, we see this advantage in reliability. The most striking effects we found, however, was in the similarity between different participants. So in this figure, I'm now plotting the correlation between the average across all other participants, the average across all participants and one participant, and the correlation between these two for each participant. So now, again, you can see that multivariate distance correlation is leading to this strong improvement in the similarity between different subjects. And that's probably the case because typically we have this misalignment of functional regions and ROIs in our subjects. They're never a perfect match. And if we have different misalignments for different participants, we might be exaggerating some of the connectivity differences between people. So this multivariate measure is able to deal with this misalignment better. Finally, we were slightly worried that all these improvements might be due to the fact that we might be picking up artifacts better with our multivariate measure. So just to be sure that we are measuring something that's close to true functional connectivity, we then also compared it to a measure of structural connectivity. And our measure of structural connectivity here was structural covariance, which is based on gray matter covariance across participants. So again, we could do this with both Pearson and distance correlation. These are the structural covariance matrices that we got. So you can see that structural covariance also contains the same similar network structure that we saw before. And now we can compare these structural covariance matrices with our functional connectivity matrices that we had before, and look at how at the correlation between them. And again, for each of the 3 sets of regions of interest that we had, we see that there is a stronger similarity between structural and functional connectivity for distance correlation than for Pearson correlation. So just to conclude, I've introduced this new measure called distance correlation, which is able to use information from all the voxels in an ROI. It measures both linear and nonlinear associations, but it cannot differentiate between positive and negative correlations. And I've shown that the connectivity estimates that we get are more reliable, more consistent across participants, and more similar to structural covariance. And in addition, we also found that it's also more robust to which set of ROIs we choose to use. So I'd like to just thank my collaborator and my funders and point out that the code for this is available on this website. And you can also find it in the neuroimage paper that we published. Thank you very much.
Thank you very much. There's time for one quick question. Yes?
A few days ago, we heard about a multivariate mutual information measure from Victor Solo. Do you know how that compares to your measure?
So yeah, I also heard that talk, and I was very interested by his measure. And we haven't actually done a correct direct comparison yet between these two multivariate approaches, but it's something I'm definitely interested in.
Great. Okay, thank you very much. Thank you. We'll move on to the last talk of the morning, and this is Hyekyoung Lee, who is talking about shape variability in the dynamics of resting state functional networks.
Thank you very much. Good morning. I'm Hyekyoung Lee from South Korea. In this presentation, I would like to talk about shape variability in the dynamics of resting state functional network and relationship with age. We have resting state fMRI data of 38 normal healthy subjects. The age of subject is varied from 20s to 70s. Each subject has 116 volumes, which measured for 6.7 minutes. Using this data, we want to know the resting state functional network in normal brain aging. But it is known that the brain network is changed over time even during the rest, so we want to know the dynamics of a resting state functional network in normal brain aging. To construct dynamic network, we simply use the sliding window analysis. We first parcellate the whole brain into 90 regions based on AAL and estimate the connectivity based on correlation method. The window size is 40 and the overlapping size is 30 points. Each window corresponds to each network. Finally, we obtain a network or subject. In this way, we apply this method to all 38 subjects, and in this way, we obtain the dynamic network. Now, the problem is, how can we find the difference between dynamic network? So, we introduce a new method based on persistent homology. The persistent homology is a new area of algebraic topology. The algebraic topology defines the topological invariants which determine the shape of network and discriminate networks. The topological invariant is called homology group, including connected component and whole. This example has one connected component and one whole. But the problem is that depending on the threshold, different number of connected components and the number of connected components and whole are different. In this example, the edge weight is defined by the distance between nodes. If we threshold it at 1.2, the edges of which weight are smaller than 1.2 are connected. So in this network has 3 connected components with no whole. If we threshold it at 1.8, the number of connected components is 1, and the number of connected whole is 1. In this way, depending on the threshold, we have different number of connected components and whole. So the persistent homology proposed to find the connected component and whole by varying threshold in this way. So let's try to count the number of connected components. At the threshold 0, there are 6 connected components, and threshold 1, 5 connected components. At the threshold 1.2, there are 3 connected components. The persistent homology also provides a visualization tool for this information. It is called barcode. The horizontal axis of barcode is the threshold. Each bar represents each connected component. So if you want to know the number of connected components at the threshold 1, you can count the number of bars at the threshold 1. So you can count 5. So the number of connected components is 5. At the threshold 1.2, there are 3 bars. So the number of connected components is 3. At the threshold 1.3, there are 2 bars. So there are 2 connected components. Each bar can be encoded into the 2-dimensional points. The first one is the starting point of bar, and the next one is the ending point of bar. So the first one, 0,1, and second and third one, 0,1.2, and next one, 0,1.3, next one, 0,1.4. These points can be plotted in the 2-dimensional space. It is called persistent diagram. In the case of a whole, it is somewhat different from the traditional network analysis because in the persistent homology, the triangle can be filled in like this. So the triangle is not a hole anymore. So in this example, there is one hole between 1.8 to 2. So we can visualize it in the persistence diagram like this. In this way, we can obtain the persistence diagram of connected components and the persistence diagram of a hole from one network. We apply this method to the brain network. We have 8 brain networks of subjects, and we estimate the persistence diagram of connected components and persistence diagram of a hole from each network. Remember that our goal is to find the difference between dynamic networks. So we need the similarity measure between persistence diagrams. So we use a persistence scale space kernel. It first performs the kernel smoothing, and it estimates PSS kernel. It is the inner product of two-dimensional space. So we can estimate the similarity between two persistence diagrams as a scalar value. So we have 8 networks, and so similarity matrix is 8 by 8. This method can be applied to the persistence diagrams of connected components and persistence diagrams of holes. So we have 2 similarity metrics. The first one is for connected components, and the second one is for hole. Based on multiple kernel technique, we linearly combine them and obtain the similarity matrix, one similarity matrix of networks in the perspective of connected component and hole. This is the similarity matrix between a network, and it is also kernel, so you can apply any machine learning technique based on kernel. So in our case, we want to find the difference between dynamic network. So if we can embed the network into the low-dimensional space, it is easier to find the difference between dynamic networks. So we apply the kernel PCA, kernel principal component analysis. The kernel principal component analysis can embed the networks into the low-dimensional space. In the low-dimensional space, the similarity between networks is approximately preserved. So we apply this method, all 38 subjects. This is the trajectory. The blue trajectory is for the younger subject, and red trajectory is for the older subject. You can find that when the subject gets older, the trajectory fluctuates more. So to quantify it, we define the shape variability by summing all distance in the trajectory. So we found that the shape variability is linearly related to the age with the significance level 0.001. So it means that the trajectory, when the subject gets older, the trajectory fluctuates more. In other words, the dynamic network fluctuates more. In conclusion, we construct the dynamic network using the sliding window analysis. We found the topological representation of the dynamic network using the persistence diagram. We measured the similarity matrix between networks in the perspective of connected components and holes. We drew the trajectory of dynamic network and estimate the shape variability. We found that the shape variability is related to the age. So in conclusion, when the subject gets older, the dynamic network fluctuates more. So thank you for your attention.
Thank you very much. We've got time for 1 or 2 questions, if there are any questions. I was wondering how you, so it's interesting to see that it fluctuates more with increased age. Why do you think that is? Well,
maybe more when the network more fluctuates more, it may I think it is related to the efficiency of networks. So if fluctuate more, it means the network is inefficient.
Right. Okay. Did you, because, oh, do we have a question there? Okay.
Hi. Yeah. Lovely talk. You looked at the zero and first order homology. Did you look at any of the higher ones, like second or third order?
Good point, but we didn't yet because computation complexity is too high, so it is difficult to estimate it.
Okay, great. We'll finish there. Thank you very much.
Thank you.